Hey, I hope it's going well. We have a very special episode of the podcast today. This was our first in-person episode. I sat down with Miroslav Gasparek, who is a PhD student at Oxford University, a fellow at the Stanford Existential Risk Initiative, the CEO and founder of a biotech startup, and a fellow at Civilization Ventures. We had a great conversation talking about control theory, a fundamental theory of synthetic biology. We spoke about co-cultures and how these are beneficial to biotechnology. We spoke a little bit about machine learning and how that's also beneficial in synthetic biology. And finally, we touched on biosecurity and the potential risks of biotechnology and how these might be mitigated. I hope you enjoy. All right. Thank you for joining me today, Miroslav. It's a pleasure to have you on the podcast. I just saw all of the things you were doing and I could not have you on the podcast. I thought it would be a shame not to have you on.
Well, Luke, I'm really glad to be here today. Thanks so much for inviting me in.
Thank you. I think a good place to start would be perhaps telling the people a little bit about yourself. What is it you do?
Right, so for everybody, I'm Miro. I'm originally from Slovakia, from a small northwestern part of Slovakia, from a small village where there was nothing even like remotely related to bioengineering all around. But there was a beautiful nature, so maybe that is something. And currently I am a PhD student in engineering science at the University of Oxford, where I'm working with Antonis Papachristou and Harrison Steele on applications of something called control theory in synthetic biology in order to improve the complex gene circuits in microbial communities. And also I've been working with Megan Palmer from the Stanford University within Stanford Existential Risk Initiative on some of the selected biosecurity and biosafety topics associated with synthetic biology. And my life dream is to become life science entrepreneur and a biotech investor and support the rise of the top biotech innovation, changing the world. And these are also some of the areas where I'm very interested about.
That was a very great introduction. I like the life goals addition into there, so people really know what you are about,
where we are, and when we are supposed or we are aiming to go, right?
yeah, perfect. So could you tell us a little bit about your research at Oxford? What is it exactly? I know it's a little bit about control theory. I've never heard of that. I've never actually researched that myself. So could you tell us a little bit about that?
Oh, and definitely, very happy to. So in these days, it's actually very interesting that when you say synthetic biology, people actually somehow feel like, oh, I know what you're talking about. Like, yeah, it's this like gene engineering, like maybe making new like Godzilla or something. I'm kidding. Or, like you probably are trying to solve some, big biological problems, but people don't understand what really control theory is, despite the fact that the control theory has been with us for ages. Like definitely, we see the first rise in maybe 18th and even earlier in 18th century, I would say, and especially with the rise of mechanical systems, electrical systems. It just became an incredibly important part of modern engineering, despite we call it like theory. And now we are trying to apply this discipline in biological systems. And if you'd like me to define what control theory is, just in a very basic level, it would be, it's about making systems behave in a way we want them to do. So it's essentially about, we have some kind of a system. It can be mechanical, electrical, biological, economic system, anything. And we want some kind of desired outputs out of this system. So, and what control theory asks is, what kind of inputs I need to put into the system so that I can get outputs out of the system I want. Obviously, it's much more complicated, and I'm very happy to talk a bit more about the details, but this is the gist of it.
Sure, yeah. So how are you applying that in bioengineering, synthetic biology?
Excellent. So I'll just speak first but on a more high level or like an abstract level, like what's the motivation for control in biology? And I'm happy to touch on what I'm working on specifically.
Yeah. Sounds
so
good.
for control in biology, biological systems are actually, they are quite interesting. They're complex. They are messy, and they are just difficult to to amend, to like manipulate. And you know a lot about this, right? So why we are applying control in biology? Well, we would like perhaps some of the bacteria we are engineering. We would like them to produce, say, some of the useful compounds only when we induce them, when we want them to, when we add some kind of inducer chemical, some kind of activator, right?
Yeah.
Now, so that's kind of the first simple control system. So I put in some concentration of an input. In synthetic biology, we would often use something like maybe L-arabinose or something like this, something called IPTG. Different chemicals, doesn't really matter. Or it can be even something sensed in the environment, and we would get something on the output, which hopefully it could just be like some fluorescing protein, as well as some pretty useful therapeutic. Perhaps we could have a gene for production of well, it wouldn't be a gene, it would be a cluster of genes of resveratrol or something like this, something really useful, commercially useful. And now, it would be pretty awesome if we were able to just come and say, so this is the input concentration I give to these cells, and they just produce this amount of certain compound at the output by activating some kind of a promoter, and then essentially starting the transcription and allowing the translation of some specific protein. However, it's usually more complex. So in the biological systems, because these cells are not always exactly acting in a predictable fashion, and there is a bunch of these challenges that we are seeing. So first of all, cells are complex. The biological systems are complex, and what I described is a very simple system, but many of the systems we would like to impact, like the global metabolic regulation, is quite complex. So it doesn't work in this straightforward way. Second, biology is noisy. So the cellular environment is not like this well-oiled machine. It's not like a pendulum that I can write these two equations and it kind of works according to them. A cell is just like you could, obviously it's not random, but it's a soup of different molecules just floating around and interacting, and maybe not interacting, and definitely not interacting as we would like them to. And this gives rise to a lot of unpredictability, and because we are often talking at low molecular counts, stochasticity. And another big challenge that we are seeing is, say, resource limitations. We don't have enough things in the cells. We don't have enough. We don't have enough kind of cellular currencies like polymerases and free ribosomes and so on that a cell could use to maybe produce our desired compound. Yeah. So it's quite potentially plausible that we want to make, say, a certain amount of the compound, and in one occasion the cell makes this amount of compound when we put certain amount of inducer in, but at the other occasion it doesn't. And the control theory, and here we're getting to this, is a very fascinating discipline, because it uses something called mostly like the feedback and even feed forward. Use the feedback control theory, where we essentially make inputs somehow depend on the outputs and on a very high level, so that we can better control, so that we can kind of sense the outputs and adjust the inputs so that the cells behaves in a way that we want it to. And the beautiful magic about this, and sorry, I shouldn't say magic perhaps, but the beautiful thing about this is that with a well-designed control system, or a biological control system, we don't need to understand everything that's going on in a cell. We have all these disturbances, perturbations, and a lot of different things. But a well-designed, well-oiled control system allows us to reduce these perturbations, despite us not fully understanding what's going on, reduce the sensitivity to modal imperfections, because we can't. I think, I'm not sure what was the name of the British mathematician who said this, but he said all the models are wrong, but some of them are useful. So there is a lot of uncertainties, and control theory in biology allows us to address all of these.
Wow, that was an incredible. Yeah, that sounds like such a useful concept, such a useful theory to apply to biology. I'm surprised it took me this long to find out about, because I've been digging around in synthetic biology for a while, so I've never heard of control theory before this. And how do you actually go about using, so you said you can sort of manipulate things in a way that you make it more controlled, you make it more consistent. How can you give examples of how you do that?
For sure. So let's start with perhaps the simplest control system that pretty much every control engineer, when you get into this area, you encounter, and that's something called negative outregulation or autorepressor. Have you heard about something like this? Autorepressor.
I don't think so, no.
Okay, so let's imagine that we have a promoter, so something that essentially allows RNA polymerase to bind to the DNA and start the process of transcription. And under this control of this promoter, we put a gene for something called TET repressor, so a protein that allows repression, that essentially allows repression of something called PTET,
right,
which is a promoter. And this promoter we use is actually PTET. So we take PTET and put a TET repressor under the control of this promoter, and next to it we say, we fuse a green fluorescent protein to it, so we get some output signal. And what we do now is that we essentially use now we use anhydrotetracycline to essentially manipulate the level of the repression of this TET to itself. So how does this look like? So what do we have? So let's imagine that we have some jump. So basally we have certain kind of leaky level of transcription and leaky level of, let's say, production of this output TET-R protein. Now, if there is more TET-R protein, what happens is that more of it binds to this promoter.
Right.
That means that less, TET-R gets made. So the more TET-R you get, the less TET-R you get. So you get somewhere to some kind of a center. And now let's imagine that suddenly maybe someone takes out just TET-R binds to something else. So it has have less TET-R protein. If you get less TET-R protein, what happens? There is a stronger expression from PTET, and you actually get more TET-R. So again, get less TET-R. So essentially, whether I have more or less of something, it gets me to this kind of stable level, this kind of somehow steady state on a high level. And now, what I've just described is called negative feedback, which means essentially when your output increases, it leads to the decrease of itself, essentially. This is called negative feedback. So it kind of it's a stabilizing thing. So because now we can see that if I perturb the system, it somehow goes back to its original level. And we can also have positive feedback, which we can see, I think, in the most famous paper one of the most famous papers of synthetic biology called genetic toggle switch, or in a switching behavior, when we can use positive feedback, for example, to induce the bistability. So, for example, if we could have this kind of we could have activator protein that activates the production of itself. And what would happen in that case, if we induce this one, the more and more protein we have, the more and more activation of itself we have, and the more and more protein gets produced. Obviously, you come to the limit of cellular resources, but this is kind of quite fast. It speeds up, dynamics, so we get some kind of a switching-like behavior.
Okay.
So I just like to get back to this negative feedback example, because negative feedback is something we often really want. I don't want to press the button and just something to kind of explode.
Sure, you need to control it a little bit.
You need to control it, exactly. And this is not only this stabilizing effect, it also has the effect of making our system robust to the perturbations, which is quite nice. I don't need to, for example, know exactly the parameters, but if I design something with a negative feedback, it will always kind of go more or less to a certain level if it's well designed. Because obviously you can design it badly, and that if it was all so easy, then all of us in our department in control theory group in biology would lose the job, which is good. It's complex, right?
Yeah.
But we would all have to be just machine learning engineers, right? But in a sense, in a sense, this is quite nice because you also get other things like speed up the response if you have this auto repressor. You get lower level of the output than if you didn't repress it, but you get fast response, which can be quite nice because I can maybe uptune it anyways.
Okay.
So this is a very simple example, like the simplest possible example of the control system we can have. And simple is good, right? Like simple is not bad. Like as simple as it gets is good. But this is not where we stop.
Right. Sure. So what is it that you are using in your research specifically at Oxford?
Right. So kind of the leitmotif or the carrying topic of my doctoral research revolves around complexity. So it's about using control theory methods and approaches along with the approaches of reinforcement learning. And I can stop there because there's a lot of analogies between machine learning sub-branch called reinforcement learning and control theory. So using the control theory approach to design and analyze complex gene circuits and I could say microbial communities. So
how complex are we talking?
Right. So and that's a very interesting question. In a sense, there's actually like people often don't think about like what does actually complexity mean? Like how would you define complexity? Like what is that? What is it?
It's a good question. I guess something difficult, something that is has a lot of layers to it, I guess.
Yeah, I know that's pretty kind of close. And you know, until I started reading more on like complex systems and nonlinear dynamics, I never really thought about it. Like we often say, like, oh, it's a complex problem, it's a complex situation, and you're like, what does this actually mean? Because like you can have a molecular complex, or some people have complexes. You know, like there's a lot of like different flavors to this. But I think you're right. It's something. I would say it's an antonym to simple when something is like simple, predictable, and easily transparent. And when we talk about complexity, sometimes something I think a good guide for us is like looking into nonlinear systems.
Right.
So what is a linear system? Well, in a linear system, and now I'll grossly simplify, we can apply the principle of superposition. So if we take a one, say one variable or like one solution of the linear equation, and another solution of the linear equation, then essentially the solution of some kind of addition of these. So we could say that the solution of the a linear combination of these solutions is the linear combination of these solutions. So essentially, all that says is something like if the solution is x, then for the linear system, the solution to problem. So if the solution to problem f of x is y, in a linear system, the solution to the f of ax plus b is a times f of x plus b. So it's kind of a linear, just moving, kind of scaling like linearly up and moving up, something like and down. Well, in nonlinear, or I like to say like in linear world, 2 plus 2 is always 4, and 2 plus 3 is 5, and so on. In nonlinear world, this is not the case.
Right.
So what are nonlinear functions? Well, anything that is not linear. So like. If I have in a dynamical system that we're looking at like x squared, sine x, like rational functions, 1 over 1 plus x, which often arises in like the molecular repression. This is our nonlinear systems. And in a nonlinear system, it's quite unpredictable because I can have like, 1 plus 1 is 2, but 1 plus 1.2 is like 10,000, or like it's just a small push out of a, certain comfortable position where the system sits, and we blow up our solution. And this happens in the nonlinear systems we are analyzing. So specifically, one of the interesting and nice areas I looked into was the system where essentially something called disturbance rejection by small RNA-based feedback circuit. Okay. That's complex. Small RNA-based antithetic feedback circuit. So to elaborate on that, essentially. I think we could say that I describe this kind of negative feedback architecture with this out-repressor, where I have something that decreases the production of itself. Well, it turns out that there are better architectures that allow us to stabilize the state of the system.
Right.
And these are actually, we could call them structurally interesting. So in biology, kind of solving out for something or like tuning up something with kind of, just by tweaking the parameters is difficult, because in biology we can't choose like, this transcription rate is going to be 1.1 or perhaps 1.12 and then 1.125. We are more like a transcription rate can be 10,000, 100,000.
Right, yeah.
So you want something that essentially makes your system structurally stable. So ideally something like the system has 20 parameters, but if I design interactions in a certain way, only 2 parameters matter. Like everything else, it doesn't matter. It will always come to the same steady state.
Right.
Which is like, it's like mind-blowing. And something like this I was looking at with like this called antithetic motif. It allows you to have this, like have a lot of parameters, but a system output only depending on a few. And I was looking at essentially to what extent action of some perturbations, in this case by small engineered RNAs, would lead to the decrease in the performance of such a system, and what would essentially happen in such a system. And again, we have, and this is very interesting problem, because one thing that we can observe in nonlinear systems is really mind-blowing. For example, what we can see in when we model is like deterministically, meaning that we assume that we have like continuous numbers of molecules and we are looking at the concentrations and everything. We can get models that show us, this system will come to the steady state.
Right.
And then you essentially come and say, how about now we assume we have a low molecular count, so like we have 10 or 12 molecules of this and that.
Yeah.
And we have essentially a non-discontinuous jumps between the numbers, so I can't have like 12 or 12.5 molecule. I can have 12, 13 or 10.
Sure, yeah.
And we do this like stochastic models. What we actually see is like what seemed to be steady state in stochastic setting. We actually see the oscillations and regular oscillations, which is totally mind-blowing.
Yeah.
Right. So it just adds to the complexity, and this was something we observed in my research. This was a project I worked on during the first year of my PhD, and afterwards, after I started looking at this like intracellular complexity, I started looking at the microbial communities, because it's just marvelous. Like we can't, be too bad to single cell, can't put too much of a burden, can't ask too much of it, and if we can, as my former supervisor from Stanford had on his, I think, Twitter handle, let the people invent the things and flourish in conjunction with the nature, so we can, bring in harmony with nature, or something like that. So don't quote me on that. But essentially, if we can bring a bunch of, partners in microbial consortia together, and let them do some meaningful things, then perhaps we can get better outputs. We can allow distribution of the labor, like this guy makes compound A, this lady makes compound B, and they all just like make together compound A, and B recombines into compound C. Like who knows? And this is actually now being done a lot in practice. But getting like different bugs like coexisting together, it's not that easy, right? They will fight for the nutrients.
Yeah.
And it's like a marriage, right? Like I don't know, I haven't been there, but it's like you probably presumably fight. You presumably fight. You have your own interest. You would kind of maybe like to help each other, but you might need some incentives for that and so on. So what we're trying to do is to, as a synthetic biologist, as a control engineers, is to engineer the interactions between these microbial consortia in such a way that, for example, their populations are stable, and if you say perturb them, so say a bunch of these bugs just dies, they still kind of recover to the original ratios or something like this. And this is allowed through a lot of different techniques, like applications of quorum sensing, toxins, and so on. So we can have bugs cooperating together, competing together, and a lot of our attention goes into understanding of these very complex systems. And one specific system and this is the class of the systems I'm incredibly passionate about, and like all you guys entrepreneurs, listen out to this one. This is a good one. So it's inducer-producer consortia. So what we observe in nature is really striking, that we often see a compound, some very useful compound for cosmetics like high hyaluronic acid, or some antibiotics, antitumor drugs, can be produced by specific microbes, bacteria, yeast, whatever. And these bugs just don't produce them in their natural state for whatever reasons, and we're like, okay, that's interesting. And now we take another bug, like a specific microorganism, we culture them together,
right?
And the production of that useful compound in this initial bug, in this producer, goes up like 50 times. Or like it actually it makes 0 and suddenly it starts making something useful.
Yeah.
And they're like why?
That's crazy.
Well, it's crazy, but when I think and what people learned is that one of the reasons for that is that a lot of these compounds are not necessarily useful if you're living in your like beautiful pampered environment with a lot of food, a lot of media, a lot of anything, and they only get produced when you activate so-called silent biosynthetic gene clusters.
Right.
And now the microbes often don't have the reason like why bother activating them. Mm-hmm. But for example, if there is another partner and it starts stealing out the, say, cofactors like iron from the environment, you're like, I better do something. So you start producing the siderophores that allow you to, take up the iron from the environment.
Wow.
And also sometimes it's different. For example, you might be able to produce a precursor, right? You get a producer that makes this precursor, but that's not really useful. Right. But when you add an inducer, it is able to take the precursor and enzymatically process it into the final product as a final antibiotic or something.
Wow.
And even more fascinatingly, you can have something like, so you take a pathogen, staph aureus, like everybody knows, like that's a bad guy, and it takes some, I don't know, was it it must have been some streptococcus holomyces or something like this. And you put them together, and obviously initially this staph aureus just kills the streptococcus, or bothers it a lot, and suddenly this streptococcus starts producing antibiotic called holomycin. Right. They're like, wow, what the hell? And then they looked into the genome of this streptococcus holomycin is. Don't quote me on the name, but whatever streptococcus species. And they learned that this species lost 1.8 kilobase megaplasmid, and then it started producing something. So it kind of became activated. Production of this voluble antibiotic became activated after you removed some gene material.
Wow.
And that is mind-blowing. And what I've been looking at is, in such a microbial consortia, with all of these different methods of action, how could we optimize the production of these voluble compounds? And why is this interesting? Well, natural microbial products market is, I think it's supposed to be $320 billion a year in 2026. Oh. There's so many things that microbes produce, and so many things that get produced in this microbial consortia. So if we can optimize the way these guys interact, that can really go a long way into helping us to produce a lot of good things more cheaply and in a more convenient fashion, and in a more harmony with the nature, in fact.
Yeah, that sounds incredible, actually. I mean, even just the idea that you can culture 2 bacteria together and all of a sudden you've got this new antibiotic that you've not seen before.
Right.
It's just...
And you want to, and often you want to, but there is something very fundamental called principle of competitive exclusion, which is... And on a very basic level says that if you kind of grow 2 bugs on the same medium with a different growth rates from a different initial growth conditions, well, one of them will outcompete the other. So that's why it's actually like co-culturing bugs is not like exactly easy. But that's where the control engineers come in, and we are here essentially to help design the interactions between these bugs, or even not necessarily the interactions between the bugs, but perhaps the design of the bioreactor and the flow rate of the nutrients or outflow of actual bacteria in the continuous still tank bioreactor, so that we can keep their populations in a certain specific predefined ratio, for example.
Okay, yeah. So it's rather than actual genetic engineering, it's more chemical engineering of how you are, the makeup of the media, what nutrients are going into the reaction, and those sorts of things.
So possibly. So there's always this kind of intrinsic control, which is like when I start tinkering with the gene circuits. That's how we call them, like this. Essentially, gene systems which essentially have some kind of these functions. And that's one kind of intrinsic control option. And then there is extrinsic control option, which is, okay, I can just, maybe put these bacteria into bioreactor and I can make them and I can just like manipulate their flow of the nutrients, and I can perhaps just manipulate the inflow and outflow of actually the bacteria in and out, and this can maybe allow me to control their populations, and I can have a mix, right? I can engineer my bacteria to be, I can put some kind of killer genes under the control of some light-sensitive promoters, and I can just essentially shine the light of a specific frequency on those bacteria, or even yeast for that matter, and essentially, induce or repress the production of the things I want. So there's a lot of different ways we can do, and my colleagues in our research group, Ting and Olivia and Simona, are looking at a lot of funky stuff. In there, absolutely.
Yeah, it sounds like there's a lot of interesting areas you could take this like research. I think so. You are more focused on the intrinsic control of the microorganisms. Is that correct? Looking at both,
because for engineers, and for a lot of engineers and control theorists, ideally we can look at the systems in general. For example, right? Like we are obviously what I like to do and what I think needs to be done is to like trying to get a good and thorough understanding of the biology, of the underlying biology. You can never understand biology well enough, but at some point you need to be able to abstract from that. And, so I'm looking both at intrinsic and extrinsic systems. Some of the principles are very similar, and some of the techniques we can use, say, nonlinear or nonlinear differential equations can be shared between both of them. That doesn't make that much of a difference. It's more about like actual maybe implementation. That's where the strong difference comes in.
Right. Okay. I think that's so much information on the control theory. I'm going to be going home and processing that for a while, I think.
I just wish people talked more about control theory, because we are not thinking about it, but it's everywhere. Like, you see, as we were speaking in this building and you came through the automatic door, right? Like, if there weren't some proper good control algorithm, the door would probably like just crazily smash. Or it would just like not even like come to the end. So there is probably a sensor somewhere, like a light sensor, looking at like, telling the computer in the door, like, hey, this is how far you are from that wall, so you better start slowing down, and essentially your kind of control objective is to stop at this point, but also not to like go crazy slow. So we need to kind of punish if it takes you say too long to like close, but you don't want to like smash the door.
Right.
And control systems are everywhere. Like we couldn't fly, like obviously the cruise control, that's big, like all these like we want to have self-driving cars, like we can't get that without the control, but there we are getting, there's something I would call a classic control. We model the systems and we're trying to control the systems. We model the linear, non-linear control of the linear system. That's kind of much easier than the control of non-linear. That's like complete wilderness. That's in a lot of man-made systems are at least to a great degree of approximation linear. Not with the biological systems, like there are pretty much no linearities. And then we are looking at this exciting area, and that's also a part of my research, which is model-free control. When we are essentially or data-driven control, and there are some great pioneers of this work, both here and around, in Oxford and beyond. But essentially, it's quite different, and it has a close connection to reinforcement learning. When we are saying like, look, I don't know how to model the system. I have no idea like how to deal with it. How about I put in some inputs into the systems? I get some outputs, like the system behaves somehow, and I'll essentially give to the system the feedback in terms of reward. So I'll kind of tell it like, I like your action or I don't like your action.
Right.
And when you look at, say, these reinforcement learning algorithms like AlphaGo, like that's what they're trained on. Like the AlphaGo, like this algorithm essentially learned to play the game. It didn't know like how to. It was just told like, we'll punish you when you lose the game.
Right.
Like, punish you. We'll just give you a high negative reward. We won't like crash the computer or something.
Right. Yeah.
And it's similar we are actually now trying to apply these algorithms in biology.
Wow.
Chris Barnes at the University College London, our collaborator, has done some marvelous job in this area where, for example, you take a bioreactor and you assume that like it can be controlled by the computer. And all you say to this computer is like, you get $2 there, you get 2 numbers out, or 3 numbers out. You get to know what the populations of the bacteria are, and you get to know like what's your reward.
Right.
And we want you to arrive to a specific to kind of stabilize these bugs that would naturally just one of them would crash, other one would stay. We want you to stabilize them at a certain predefined ratio. Learn it.
Okay.
And essentially after a couple of iterations and episodes, it can learn.
Wow.
But obviously this has a lot of drawbacks. Like there's actually something I feel sometimes like, like reinforcement learning or machine learning, like that's this like coming of the savior and like end of the world and we are going to control everything like that. Like, the truth is that 95% of the systems in this world, man-made systems, we can perfectly find control of it, like the most classic, boring controllers, which are called PID controllers, proportional integral derivative controllers. Every second year undergrad here at Oxford, that comes to like our control theory classes, learns this easy. It works. Like, don't reinvent the wheel. But there is this like 5% of like juicy, lucrative situations: self-driving car, optimal chemotherapy tree selection, optimal treatment, nonlinear biological systems, all like economics. That's a large. Like all of these things.
Yeah.
They are really juicy, really lucrative. That's where we need like all of these like complex control theory and Lyapunov functions and all the weird and not weird like theorems and reinforcement learning, for example.
Yeah.
Because it has a lot of challenges, if you take something model free.
Yeah. That's incredible that you're applying those reinforcement learning algorithms to the biological systems. Is that like, as in you're applying it in the model phase or in the actual, physical growing of these bacteria?
Well, we want to get there, certainly, at some point. But what I'm, for example, doing is kind of, I know, in the reinforcement learning paradigm, you usually kind of split the system into two things. You have agent and you have an environment.
Right.
So agent is your kind of algorithm that learns, and what agent does is agent gets kind of the information about the state of the environment, about the next state. So kind of at a state at a time t-1, state at a time t, and it applies some action to the environment and it receives some reward after the action.
Right.
So essentially what reinforcement learning agent gets is like this kind of tuple of previous state, current state, reward, and action, and what it outputs is the action into the environment. It kind of modifies the environment with which it interacts. For example, you can have a reinforcement learning agent, which is a car, and like it applies some gas to the throttle, and the environment is essentially the thing that takes the action and what it outputs is the reward.
Right.
And it outputs is the states. So, and it's this kind of a loop. So, and the point is now like the agent, that's always kind of, usually always that's the algorithm. That can be the robot, that's an algorithm, whatever.
Yeah.
And the environment, it can be something physical, it could be a bioreactor, or it can be a model of a bioreactor. Initially that's equally good. So far I'm working with the models, but at some point I'm sure that my supervisor, Harrison, will hopefully get to the stage where we will implement something in the lab, and that would be just marvelous. So, yeah.
Be exciting to see that. I hope that goes ahead.
easier to iterate, in silico in computers. Then that's the advantage. That's the advantage of a lot of us who are working like computational side of the PhD. Like you run the simulation, and usually running that, unless you're like doing this like some crazy Amazon Web Services mega clusters, it's much easier to run something like this.
Right.
And like go to the lab and like, oh, I screwed up my culture. Oh, like, yeah, oh, damn. Like, I work in a lab as well, right?
Yeah.
So I know the feeling when you just contaminate your cells and you can start all over. So
Yeah. I'm conscious of the time. I wanted to touch a little bit about what you do at Stanford.
Yeah, let's get into
the
it.
Existential Risk Institute, is it called?
Yeah, it's Stanford Existential Risk Initiative. And yeah, it's really exciting. So just to step back, what we talked about until now is, very technical, very engineering and very mathematical part of synthetic biology, albeit incredibly, practical with strong implications. And I've been always really fascinated, and that was mostly the interest of my former supervisor from Stanford, Drew Endy, about kind of wider societal and political and whatnot applications of synthetic biology. And I've just been, incredibly honored and pleased that I could have worked with Megan Palmer, Professor Megan Palmer from Biopolicy and Practices Lab at the Stanford University.
Right.
And working. So what Megan is a specialist on is like the policy and regulation of synthetic biology and thinking about the wider societal implications. Because there is one thing, if like can we do something, like that's what engineers are solving, like enable us to do something, and then there are policy questions that need to be answered, which is like, should we do this, and how to do this so that it has maximum benefits and minimum downsides to ourselves and to our surrounding.
Right.
So what I've been working on with the Megan is essentially the biosecurity and biosafety considerations related to these microbial communities. So I've been really glad that I could have worked with Antonis and Harry on this kind of technical aspects of microbial communities.
Cool.
But with Megan, I've been looking at like, so what are the existential risks that microbial communities can bring us? Maybe not necessarily like existential, like it will maybe, it's so it might not like, completely destroy the humankind, but more of a looking onto like how can we misuse, or how can the microbial communities be misused. And there are biosecurity, considerations and biosafety considerations. And just to explain, so biosafety is associated with like we could call it like unintentional, avoiding unintentional improper use, right? If you accidentally spill out, I don't know, anthrax in your lab, I hope folks, you don't just carry around anthrax spores in your lab. But if you do that accidentally, that's a biosafety incident. Biosecurity incident, this is like, let's go take this anthrax spores, let's make them 10,000 times more virulent, and let's put them to, I don't know, let's say, St. John's College lunch of your unfavorite professor or something like that. So that's a biosecurity incident. That's where we want to get you. And we need to look essentially at both of these things. Biosafety and biosecurity, both are incredibly important, and try to develop both policy measures and regulatory measures on one hand, and technical measures on the other hand, to avoid the incidents in the case of biosafety, and even wider incidents like extinction of the species, if we just like allow our super engineered superbugs to colonize certain ecological niches, and how to avoid like bad actors doing bad and kind of stupid things, like engineering pandemics and just harming the people and harming the planet.
Yeah, it's so important. One of the reasons we actually started with this podcast was to talk more about this sort of biosafety area, because we felt like it was just not out there that much. And I guess that was maybe my lack of finding it, I guess there are.
I think you're totally right. You shouldn't feel bad. I think that there is a wrong framing of the popular culture with like synthetic biology, and like genetic engineering, because people often like, you even have like a lot of these, signs like GMO free. I'm like, are you serious? Like, but that's not like, I'm not like, we should not be angry about the people or anything like that. It's the way how the modern culture portrays synthetic biology, and I feel it goes to the extremes. It's either like this, paradise, like with synthetic biology will cause us to live in this like paradise where we can like grow anything on a trees, and I don't know, we'll just all be like using Bitcoin and using, I don't know, synthetic biology to supply everything, and we will colonize the Venus with our super engineered plants. I mean, maybe, but this is a rosy future. And then we've got like this other one, like, oh my god, synthetic biology, they're like, we need to burn them like the witches, they are going to destroy this world. They made, I don't know, they will make engineered pandemics, and they will all bring us to that. And yeah, so there is a certain middle ground which should be like, this is a serious engineering discipline and it can have immense benefit for the humankind. However, as pretty much everything in this world, from nuclear energy to religion, any kind of religion can be misused. So how about we think how to avoid misusing this and misappropriating it, and use or like apply engineering biology principles to do good things and to improve our world.
Very well put. I think that's a great place to end the podcast. Thank you very much for joining me today. It's been incredible. Thank you very much.
Luke, thank you so much. This was a great pleasure. Thank you.
Cheers. And that's it for today's episode with Miroslav Gasparek. As always, if you enjoyed the show, remember to share it with your friends, share it on social media, give it a like and a rating. That would help us a lot. You can follow us on social media to stay updated with future episodes and the clips of the podcast. Thanks for listening. I hope you enjoyed the show.